Hi, I'm Carolyn. I'm so glad to have the chance to work with an excellent group of scientists in Hong Kong to improve our understanding of immunity to influenza. Immune responses to influenza virus infection and vaccination involve antibodies that either neutralize a virus or engage other arms of the immune system, and involves the presentation of virus peptides to CD8 killer and CD4 helper T cells. These CD4 helper T cells secrete cytokines and support antibody production. In one recent study, we have assessed virus-specific antiviral cytokine production in CD4 and CD8 T cells after influenza vaccination. We stimulated whole cell samples with whole viruses grown in our P2+ facilities and stained them directly ex vivo for surface and intracellular cytokine markers, and then tested them by flow cytometry. We found that vaccination increased the polyfunctional, aka high-quality, helper T cell response, but that repeated vaccination was associated with attenuated T cell responses.
Hi, I'm Isaac. As Carolyn said, antibodies are an important part of the immune response to influenza virus infection and vaccination, and that's my lab and my area of focus. We are particularly interested in infection and vaccination responses in older adults. I have been investigating the consequences of repeated influenza vaccination in older adults on the antibody responses. Preliminary data showed that despite being repeatedly vaccinated with the same antigen, some individuals still do not respond well. Our recent data suggests that the immune response could be making antibodies that target other viral proteins. I will be looking at antibody responses to different strains or targets and investigating the underlying cellular changes associated with weaker responses. This will allow us to design better vaccines against rapidly evolving viruses.
Hi, I'm Katrina. I have been monitoring the effectiveness of influenza vaccines each year in Hong Kong. Influenza had disappeared from Hong Kong during the COVID-19 pandemic, but returned in early 2023. Similar to before the COVID-19 pandemic, the influenza vaccine has generally worked better against the influenza A H1N1 than A H3N2. And my research focus is on trying to understand why that's the case. These studies inform local vaccine policy and also contribute to global strain selection for the next season's influenza vaccine. We would like to thank the Research Grants Council for funding to support this research.